I actually was up all of last night preparing this talk because I was like, okay, what am I going to say? And so I got a couple hours of sleep. If I'm a little sleepy and if the talk isn't quite polished, that's because I've been thinking very hard about what to say. And because Jerry sent me a very hard task, he said, explain how you think about neuroscience, and the project you're doing to a general audience, to an average audience. And of course the problem is that the average audience does not exist. Experts but not an expert. The problem is that these distributions don't have any means, and we are in the tails of this. So with that apology, and I think Artem asked me if the person on the street would understand this talk. So this is very challenging. I don't know if I'll be able to really do this, but I invite you to just share some of my thoughts here. And without further ado, I will start with a little piece of trivium. Who says this? Who said this? Experience shows that nerve force is generated and set free whenever the cerebrospinal system is excited. The direction which the nerve force follows is necessarily determined by the lines of connection between the nerve cells with each other and with various parts of the body. But the direction is likewise influenced by habit, inasmuch as nerve force passes readily along accustomed channels. If you know who wrote this, please raise your hands. Charles Darwin, neuroscientist. So I highly recommend this book, which was well ahead of its time. Darwin made a psychological questionnaire which he circulated to individuals across the globe, collected the results, and also studied animals on his own. He used to breed animals himself. And he, this was 100 years ahead of its time because people kind of ignored this work for the next 100 years or so before picking it up again. He establishes two very important things. One is the invariance of emotional expressions across cultures. So he says, I have endeavored to show in considerable detail that all the chief expressions by man are the same throughout the world. And what he establishes, very importantly, is the innateness of emotions as opposed to them being culturally learned. And this was caused a huge conflict. As I'm sure you know, there were culture wars around this issue. In modern parlance, the affective circuitry in the brain, which is encoded in the genome, underlies our emotions. The second very important thing that he establishes is homologies between emotional expression in human and non-human animals. It makes sense. Darwin is thinking about evolution, but this is of critical importance to us. Affective disorders, which underlie perhaps the bulk of a neuropsychiatric illness, can be fruitfully studied in animal models due to the strong conservation of affective systems across mammals. So, this beautiful book, highly recommend, and he establishes these two things that, I couldn't say any better. So I would like to point to this book by Jaak Panksepp called Affective Neuroscience. He may have coined the term where he essentially studies this kind of homologous circuitry in animal brains that underlie emotions. And the idea that he has, and I do not want to say that it is his original idea, but it is articulated by him in this place. Took this diagram out of his book. The idea is that there are specific and distinct, although somewhat overlapping neuronal subcircuits. So here's four of circuits that he or systems that he draws, and the idea is that stimulating these circuits solicits specific affective behaviors. So there are famous experiments going back to Walter Cannon, whereby stimulating a part of the cat brain, the periaqueductal gray, he gets the cat to show sham rage. And this was very important because William James, so there's a James Lang theory of emotions, which essentially says that emotions are when the brain senses the viscera. You see a tiger, your heart starts pounding fast, your stomach churns, and you feel that in your brain, and that's what an emotion is. And Walter Cannon And Cannon-Bard theory kind of debunks that and says, no, it actually goes the other way around because you can stimulate the brain and can generate these emotional expressions. And of course, it's a feedback system. Darwin, interestingly, is aware of this. I'm not going to cite him, but you should read him. It's amazing to see how he talks about this. So there's a feedback system, but the idea is that there are these somewhat distinct circuits, which can be kind of listed. You can pick your nomenclature. They can be studied somewhat in isolation. And these are mesocircuits. So this is where this notion of the mesocircuit that Jerry so kindly kind of delineated for you come in. And one of the reasons why I'm interested in understanding the mesocircuit of the mammalian brain, because one of the great questions that has been there since the Greeks and the ancient Hindus is how do our emotions work, how do we feel, and so on and so forth.
Question: is this 4 out of thousands or
4 out of 7 that he defines 7, and here are 4 out of 7. And seeking system, other people kind of call the reward system, fear, panic, it's also called attachment, rage is called anger. I don't know, the terminology differs. The nomenclature, you can open Bharata's Bharatnatyam, which is the classical Indian text on drama, and you'll find a categorization of emotions, right? And also, description of what facial expressions go with it, how to, method acting course. But nomenclature varies. I do not think, Yock's system is perfect. These things will need further discussion.
That wasn't my question. My question is, are we talking about
a few
or thousands?
A few. The idea is that it's a few.
It seemed awfully surprising.
Well, there are only a few.
Well, exhausted, but anyway.
There are only a few major behaviors, if you want, for animal behaviors: nutrition, reproduction, defense in the individual and social context, and to manage these behaviors, you sort of have these systems. This is a oversimplification. The brain's a very complicated thing, but as theorists, we want to simplify it. It's probably not two. It's probably not just reward and punishment, which is probably a gross oversimplification of affect. So, following Jerry's exhortation, I have generated a 2.5 slide primer for the nervous system. So those of you who work in neuroscience will be annoyed or bored. Others may or may not be mystified. I don't know. This is impossible to do, but I thought I'd do it anyway. And this is also a personal perspective, because everybody has their own theories about how the brain works. In fact, nobody has theories about how the liver works, but everybody has theories about how the brain works. You can ask any person on the street and they'll tell you. The nervous system has a genetically encoded architecture which is unfolded through a developmental process subject to environmental input. So it has both phenotypic and adult plasticity. It stores programs for core behaviors, nutrition, reproduction, defense, in individual or group settings, requiring locomotion. So if you're a plant, you sit in one place, you make your food, you don't need a nervous system. If you're an animal, you have to go find your food, so you have to avoid obstacles, and you need to see where you're going, and you develop a nervous system. And Rolf Linas gives the example of this sea squirt, which has a sessile face where it moves around, has a little ganglion, a little brain, and then it decides to anchor itself, and no longer needs to move around, digests its brain, and so brains have got to it. Moving around, not really giving PowerPoint presentations, right? Locomotor and core behaviors are subjected to feedback control based on sensing the environment. The nervous system, so I'm kind of taking what will be familiar to neuroscientists in the audience as an ethological perspective rather than more of a behaviorist perspective. The nervous system consists, as Jerry has pointed out, of a network of neurons connected to each other and to sensors and effectors, both internal and external. There's an external environment and an internal environment. There's the visceral environment. Now, as Jerry has pointed out, this is quite remarkable. Neurons are the longest cells in the body. You've got neurons going down from your spinal cord all the way down to your toe, from your brain all the way down to your spinal cord within the brain. Huge processes, right? Remarkable. And they coordinate the dynamics of the whole organism. It's not local control. It's a command economy. It's commanded from the head ganglion. You lose your connection to the spinal cord, your arm doesn't behave by itself, right? Neurons exhibit specialized electrical signaling with all or none action potentials that propagate rapidly along axons. Individual neurons, as well as networks of neurons, exhibit spontaneous as well as induced activity. If you're a neuron in globus pallidus, you're sitting there firing away. You don't need any external input. If there are other neurons which are not firing away spontaneously quite so often, but there is just intrinsic activity, vacuum activity, and then there's also induced activity. Both are important for nervous system behavior. Now, this is partly what makes it very complex. Neurons exhibit complex and specialized intercellular chemical signaling via synapses. So neurotransmitters fall into, if you want, broad classes with increasing specificity. And I've only given examples, right? So there are the amino acids, glutamate, GABA. There are a few more. These are the major neurotransmitters. They are. There's one with a positive sign, one with a negative sign. So these are all transmitters. They're released by axons, but some of them get called modulators, and the so-called biogenic amines. So there's about order 10 of these. So if you start counting numbers, there's this group which is related to arousal, metabolic arousal, sensory arousal, motor arousal, in kind of along a little axis in the brain. There's serotonin, which is not so much arousal as kind of quieting the system down. These are all words, kind of folk theories that I'm throwing at you to some extent. There are other major transmitters, acetylcholine gases such as NO. So these are modulators in the sense that they modulate the activity of the major transmitters. And then there are the peptides, which have even higher levels of specificity, and there's many more of them, right? So there's less specificity, fewer with less specificity, many with more specificity. Order 100. I don't know, maybe more than that. For example, the famous beta endorphins, the pleasure molecule you get by running, and so on and so forth. Although there's controversy about that. Stress, corticotropin releasing factor. And these have both peripheral and central roles. That's interesting. So, for example, angiotensin has a peripheral role. It causes water retention in the kidney. It's got a central role in the brain, which promotes thirst. Now, there's further complexity which is added because for each of these transmitters, there's an array of receptors. And depending on which receptor you have, a transmitter may have completely different actions. Right. So on the one hand, it's very simple. You have this axon which has got electrical action potentials, and you should think for a minute about how remarkable that is, because inside a neuron, there's all these genes being expressed. Its state is so very complicated, tens of thousands of different state variables probably. But the Hodgkin-Huxley equation, for example, keeps track of only 4 state variables: the voltage, or order 4, order 10 state variables. And you can write down closed form ordinary differential equations, partial differential equations, to describe the change of those state variables. This is really remarkable, right? And that has caused electrophysiology to be a quantitative subject. But then sitting on top of that is this sort of diversity of transmitters and receptors, which make life very complicated. It makes it easy to try to create drug molecules, but makes it difficult to understand the brain. There do exist kind of macro architectural principles of mammalian central nervous system circuitry. For example, since the early 1800s, there's been known this so-called Bell-Magendie law that ventral is motor, dorsal is sensory. It's true in the spinal cord. You can even look at the cerebral hemispheres and make this sort of connection. Or exteroception, your external senses and locomotion, your somatic stuff, that's the thalamus and the neocortex. Whereas interoception, sensing the interior of the body, regulating that's sort of the hypothalamus paralimbic system. So there's some broad organizational principles of the nervous system. It's not a tabula rasa, a bunch of neurons in a dish which are punished until they behave properly, right? So the nervous system is structurally complex. So I put up this little video of a zoom in of a brain section that has been stained with Nissl and myelin. One more time, zooming in into the Nissl and myelin. This is not a great myelin stain. The myelin stain stains myelinated axons. This is kind of a fuzzy stain. Please go later look at the one that's outside. But all I really wanted to point to is the structural complexity. But I have a few more slides to do that. Now this slide. So this is Winfried Denk, Moritz Helmschutter, and this is the work that came out recently that I think Jerry was referring to. I'm not sure if it's the same paper, but this is just a kind of fly through EM fly through a piece of neuropil in retina, and this is just meant to show you that there's a lot of ultrastructural detail. What you're seeing are pieces of neurons. They're all tangled up in a little spaghetti. I don't want you to, look at this in great detail. All I want to emphasize to you is when you get down sort of to this level, there's a lot of ultrastructural detail to take care of, right? So nervous system is very complex. You kind of got that. But people keep talking about this complexity. So, however, this is not disorganized complexity. This is very different kind of complexity than you will find in a gas or in turbulence, for that matter. This complexity is organized. You find nuclei, lamina, fiber bundles, also more diffuse reticular structures, and this is classical neuroanatomy. This is a section of the Allen Reference Atlas created by Hongwei Dong. What you see on the left is a Nissl-stained section. So there's a microscope outside. I welcome you to look afterwards at Nissl-stained and myelin-stained sections yourself of the mouse brain, any of you who are interested. It just shows cell bodies as dots. And you can see, there are clusters. It's not a homogeneous thing. And in fact, by using both cytoarchitectural and the response to external stimuli or other means, people have, and segregating projection patterns, people have broken up the brain into these regions. So neuroanatomists have done this. You can open up atlases and look at this. Several hundred regions. And the wiring patterns, if you just look at the myelinated fibers, are also quite organized. It's not a random soup of any sort. It's not living in some high-dimensional neural network space where you can't discern any structure in the three-dimensional space that it is actually embedded in. You can discern structure in three-dimensional space. Okay, that's the corpus callosum. Various other fibers. Again, these are the myelinated fibers, not all the fibers. These you can't really see, but these little dots are fiber bundles that are running perpendicular to the section that you're seeing. Now, I think what motivated me to take on my project, Jerry, what's my time scale? How long should I be speaking till? Quarter of six. Okay, good. I have time. Okay, so single neuron biophysics has been well explored. And that, I think, is due to this remarkable phenomenon, which we don't really think of as remarkable, but it is remarkable that you've got this closed form differential equations describing single neuron biophysics. I think that's a non-trivial fact, really, about the nervous system. We know a lot about synapses, and microcircuits have been studied a fair bit. And we're also looking at the circuits that live inside a neuron, so the intracellular kind of genetic regulatory networks, protein networks. But there's kind of this relative knowledge gap at the whole brain level. And what I mean by that is the following. So if you look at the peripheral nervous system, there we have a fairly good system diagram of the parts and their connections. Okay? And this is very central to medicine. You go to a hospital, this knowledge is helping us, a lot of us, live. I mean, when we get into accidents or something happens, right? We don't have anything analogous to this for the brain, for the central nervous system, right? So that's what I mean when I say that there's kind of this missing level of knowledge. this is known. Francis Crick and Ted Jones wrote this article in the early 90s talking about the backwardness of human neuroanatomy. Human neuroanatomy is even worse because in the animals you can do injections, let the neuron transport those tracers, sacrifice the animal, take out the brain, and look. In humans, that experiment is ethically challenged. It cannot be done. And as a result, we are stuck with about a century-old data set, which comes from gross dissections, myelin stains, and so on and so forth. And much has been made of functional connectivity, quote unquote, which is a misnomer. It is really the correlations of activity or diffusion tensor imaging and so on and so forth. But that won't really reveal to us this, actual neuronal connectivity pattern. So, big problem. But what shocked me was not the fact that we didn't know the human brain in such great detail, but we don't know the rat brain in great detail in terms of this level of connectivity. And this I found by attending a talk by Larry Swanson many years ago, where he had put up this matrix, which he had compiled with his postdoc, Michal Bota. So let me explain to you what you see. So they took order 400 papers, many of which Larry himself has written, and curated information from them about whether one brain region connects to another brain region. So you have to talk about a number of brain regions. So they have divided up the brain into about, and by brain I mean rat brain in this case, into about 500 compartments. And wherever you see a gray or a colored dot in this matrix, there is evidence, either positive or negative, about the existence of connections between those regions. Where you see gray, there is no evidence. And 90% of that matrix is empty in this curated database that Larry created. And of course, he has not read every paper. We cannot estimate how much, we don't know how much we don't know about the rat brain connectivity. Yes?
Does connectedness mean oriented connectedness?
Correct.
So this matrix is not symmetric.
It is not symmetric. And there is an additional complexity. Matrices do not capture the full architecture of the nervous system because of collateralization. So I could have, of this mesocircuit because of collateralization. I could have nucleus A talk to nucleus B and nucleus C separately using separate projections, or using one axon that then bifurcates. So that information is not captured in this connectivity matrix. So this is not by any means a full representation, right? But even so, I found it shocking that...
But is there any sense to say how sparse this matrix would be if you knew all the connections?
No, we don't have that knowledge either. It would be not fully connected. So
well, certainly not fully connected. So the question is it sparse? It could be a quarter full or half full?
Don't know.
No gut feeling?
My gut feeling is that at this level of connectivity, each brain region is, well, from what we know, those connections which have been studied, each region can talk to dozens, order 10 other regions. And I think the difficulty with this sort of matrix representation is we are really talking about space, and you're parcellating space. Sometimes there are clean parcels, sometimes there are not. But it'll be more sparse than not. I'm making a rhetorical point. So maybe the situation is a little better, than this kind of bleak 90% number would show. But this information is also scattered among papers, which we can no longer read. I can no longer read papers. There's too many of them being published. So I've actually learned more neuroanatomy simply from running this experimental project and collecting an integrated data set.
What are the two dark bands? Can you say?
Two dark bands are, so injections which then show that, so each vertical or horizontal line is an injection somewhere, and you can tell that this part did not project to most of the brain. So that's a dark band.
By connection, do you mean a single neuron going all the way, or A connects to B?
No, a single neuron going all the way.
Okay, so a multiple thing doesn't count.
Not in this. So if you iterate this matrix, of course, it'll soon become very full.
So, since it's a directed graph, if you just find the strongly connected components, they break up into one big strongly connected?
Great idea. I mean, but I need the graph first, right? So
Right.
that's exactly the kind of questions which has been asked in bacterial metabolism, for example, very nicely. So you can take the kind of stoichiometry matrix for bacterial metabolism and break it up into block diagonal structure. Surely our brain also does that, and we can make guesses about it. But I was like, well, why are we missing this data set? And I was like, because it's too hard, because it's too expensive. Then I started looking into it and I decided, no, it can actually be done today. And I'll tell you maybe what some of the bottlenecks were before. But, you all know that there is this contrast with the availability of the full genomes of many, many species, right? So I invite you to take a look through this paper, if you like. It came out of process of having several meetings with a large group of people. I learned a lot. I circulated it as a link, and the important thing is, so what we proposed was that there should be coordinated efforts. I don't want to draw facile comparisons with the Genome Project because I don't think that is merited. But nevertheless, I think that we do need concerted efforts. This is one thing where we are suffering from, I think, a tragedy of commons, because this is the kind of thing that no individual laboratory can really do easily very well. There's sort of centralization of resources necessary, and also multiple species have to be studied so that we don't keep studying just one species. So we need some coordinated effort to map out neuroanatomical connectivity at the whole brain level, and that I think is only feasible for the larger animals. If you're not the worm, it's only feasible sort of at this mesoscopic level. Yeah.
So who's going to determine what the units are at the mesoscopic scale?
It's an empirical question. So
Well, I mean, you gave a nice picture, you showed a nice diagram of the peripheral nervous system, and the reason that we know all those things is we know the endpoint is, right? You sever a certain peripheral nerve, something is or is not innervated, and you're done. The units are in the brain are not so well defined.
there are some data-driven ways in which we can go about this, right? So how did classical neuroanatomists did this? Because they did draw these little boundaries, which they quarrel over. I mean, there are boundaries which are essentially like India and Pakistan. Somebody draws a boundary here, another person draws a boundary here, and they cannot be in the same room at the same time. They're looking at the same evidence. But
So I'm just wondering, is that
some of the boundaries are clear.
You view that as done?
It's not done, but good progress has been made. How would we do it? We would look at I'll show you an example of a data-driven way in a little bit later in my talk. Cytoarchitectonics, gene expression patterns, projection patterns, mapping of responses to external stimuli. There's a whole bunch of things which one can do. We have good preliminary evidence that this is going to be the case, very good preliminary evidence. It can be made more quantitative. Okay, so we've been kind of discussing this. I think it's impractical to define connectivity atlas at an individual synaptic level. It has been proposed because the idea is that, if you knew every synapse, then you could determine these other levels. But the problem is there's individual variation. Things change as a function of time. You don't have the same set of synapses, today and a few months or a year from now. Very crude first step, the cytoarchitectonically defined areas would in atlases would correspond to this network. But I could caution you that you can't always draw boundaries. Sometimes you can draw clean boundaries, sometimes there are gradations, right? The point is that there are spatial correlations. That it's not one neuron is not that different from its neighboring neuron as long as you account for cell type and things like that. There is kind of some correlation structure in the brain. The brain's a highly structured correlated place. So even if it doesn't break up into parcels, there will be some low-dimensional representation of the structure, some of which we know, some of which will have to be data-driven and discovered. Segregating projection patterns. I think I've belabored this for a while. I'm not going to talk about this anymore. I want to not get labeled as the mesoscopic guy. I mean, I'm just saying that this is like the first step you take. But I am fully aware, that there's a multiplicity of cell types. I have the problem of attacking many problems at once, but even I discipline myself sometimes, one at a time. So I think this is the level we can go for now, but we should fill in, the more refined information. And very important subtlety to keep in mind, which you already pointed out to you, is collaterals. Neurons are trees. They should not be thought of, point to point. Okay. All right. So since there's a lot of theorist friends I have in the audience, I'm going to take a three-slide digression and indulge in a bit of philosophizing, and pardon me for doing this, but I thought you would be interested in hearing my thoughts and I'd be interested in hearing yours. Why is it important to study this whole brain level of architecture? So what I wanted to do is to lay out for you some prevalent theoretical dogmas, perspectives, all of which have validity, but I'm just trying to lay them out. So there's a chemical soup point of view. The brain's a soup of chemicals, disbalances in this soup cause this disease, fix those disbalances. This really focuses in on the transmitters and receptors, right? But the transmitter-receptor systems only make sense in the circuit context. You can't really talk about the transmitters and receptors without the circuit context. So it has some truth, but not the whole truth, the chemical soup model. There's a place theory of the brain. It's a time-honored theory. Used to be called phrenology. Nowadays, phrenology is not such a dirty word anymore. But it is true that there are specialized brain regions, which is known from lesions, more recently from brain imaging. However, the importance of thinking about circuits is made clear by the instance of deep brain stimulation in Parkinson's disease. So dopaminergic neurons degenerate in substantia nigra, that's place one. However, you fix it by stimulating or doing surgical intervention in place two, which is subthalamic nucleus or globus pallidus. These are all connected. You have to understand the circuit context to understand why this therapy works, right? So the place theory in and of itself is not going to solve the whole problem. And there's this more kind of subtle thing that's going on, which I call, because I do come from a theoretical physics background, I recognize it very vividly. There's sort of a physics-style theorizing that kind of assumes homogeneity and simplicity. And if you take the homogeneous and simple point of view, you're not interested in the macroscopic structure. You're only interested in kind of local connections. And maybe somewhat unfairly, and for polemical purposes, I will put tabula rasa learning and canonical cortical columns in this box of physics-inspired theorizing. And so the idea is that there are these kind of universal units that are replicated through cortex, and all we have to do is to understand the input-output properties of those units. We will understand how the brain works, so we don't really need to understand this mesocircuit, or it's kind of secondary to some extent. Or supposing all you really needed to know was the rules of synaptic plasticity, and the brain wired itself. And you didn't really have to pay close attention to the developmental programs or details of anatomical architecture. Again, you don't care about this whole brain level of architecture. If you adopt either of these 2 points of view, your research program, my research program does not make much sense to you. Like, why are you doing that, right? I kind of, have been thinking about this for a while, and then I realized, after reading Poincaré that, other people have been thinking about it too. Poincaré says, it is therefore thanks to the approximate homogeneity of the matter studied by physicists that mathematical physics came into existence. In the natural sciences, the following conditions are no longer to be found: homogeneity, relative independence of remote parts, long-range connections, simplicity of elementary fact, and that is why the student of natural science is compelled to have recourse to other modes of generalization. So there's this book called Sciences of the Artificial, which I highly recommend, by Herbert Simon. And so I think, engineering is thought of as a blue-collar discipline. You make things. But it has to become a white-collar discipline where you really have to think about biological systems from an engineering perspective. Physicists never have to do that. The Earth doesn't go around the sun for a particular purpose. You don't have to think about selection of planetary motion. But in biology, you have to do that. And it doesn't really matter that there is not an intentional engineer. There's an engineering process, if you want, selection. And that kind of influences how you theorize. So in physics, the basic laws, if you want, are symmetry principles. So the system is invariant under the action of suitable transformation groups. So I may have lost some of the audience here. I apologize for that. I will return after this slide. So in physics, physical systems fall into kind of universal classes which all respect the same symmetry groups, right? So that's what physicists know and love. In biology, there is no invariance with respect to physical symmetries. Your body doesn't look like the same thing from one part to the other, except in some trivial sense. there is no useful kind of translational invariance in space and scale. I don't want to make a generic statement. Biology always comes up with exceptions. But in general, it is, these are not the interesting symmetries. However, in engineering, there is also a notion of universality. You all know it. For example, the universal Turing machine. So the idea is that as long as machines share some core architectural features, they can exhibit the same behavior. And the behavior is all that the environment cares about. So it's the invariance of the interface. So it's a different kind of invariance. But there are also universality classes in biology. It differs from the kind of universality classes we are used to thinking of in physics. But how are we going to go about empirically studying it? And the one key biological tool here, which we almost don't use as much as we should, at least from a theoretical perspective, I think this is the most important question almost to make kind of deep theoretical insights, which is convergent evolution. So what aspects of nervous system architecture, if any, have evolved repeatedly and independently? And this is an empirical way of separating essential from non-essential features of the nervous system. If there are core architectural features which you need sort of for the function of the system, so to speak, it will have been reinvented by biology multiple times. And if we can, get at across taxa, if we can get at the system architecture, I posit you can do this at the mesocircuit level, then we will have made progress. I think I've spoken way too long about that. I will stop there. I can discuss that with you later. But so my main take-home message from all of that was that my motivation for studying these brain-wide circuits is really evolution. I think that neuropsychiatric disorders are also very important motivation. I talked about emotions in the beginning, but I just wanted to throw in this sort of evolutionary theoretical kind of perspective. So neuroanatomical methods have evolved over time. Actually, one method that's missing on this slide, very important, is Golgi's method of staining individual neurons, which Cajal made excellent use of. The method that we are making use of in the project that we are proposing is neuronal tracers or using viruses. Here's an enabling technology. In 1989, believe it or not, a terabyte of data cost tens of millions of dollars. The projection is for 2020, $3 a terabyte. I don't know if that, humongous drop in price. Why does this matter? Well, the mouse brain at a micron resolution, which is optical microscopy, is about a teravoxel. So I have several mouse brains stored now in my lab. It would have cost billions of dollars just in the beginning of the decade of the brain. It was too early, right? A human brain is a petavoxels. So right now, petavoxel, that's getting to be a little difficult for research budgets. So genomes are a gigabyte. Sorry? I'm sorry. A voxel is a volume pixel. So it's if you take the brain and you cut it up into little cubes, 1 micron on the side, that's a voxel. So the mouse brain is about 1 tera, 1 micron cube voxels. The human brain is 1 peta, 1 micron. Here's a picturesque way I've come up with thinking about this. If you take an axon, a thin axon, like 1/10 of a millimeter, if you want to magnify it so that you can see it by the bare eye, that's about an axon is 1/10 of a micron. If you want to blow it up so you can see it by the bare eye, that's about 1/10 of a millimeter. So that's a factor of 1,000 blow up. You take the mouse brain, you blow it up by a factor of 1,000, that's 10 meters. That's about a 3-story building. Take a human brain, blow it up that much, that's a football field, that's 30 meters. So in thinking about the complexity of the brain, you have to think about a football field, sized cubed, and then you can barely see the individual processes, right? It's Amazon rainforest in there. Okay. So following on the footsteps of the Allen Institute, we have made this high throughput anatomical pipeline. We have tried to industrialize neuroanatomy. We are now able to process multiple brains a day. Usually that's whole PhD thesis in the context of doing classical neuroanatomy. But those sorts of kind of economies of scale or speed ups are familiar to people who have worked with genomes. So we are trying to bring that to neuroanatomy. And the Allen Institute, as I said before, really pioneered this. But I believe that it needs to be democratized because science cannot be done with n equal to 1. For example, the sagittal gene expression data set from the Allen Institute is n equal to 1. 1 mouse one gene, and no, they don't have any current plans to replicate that data set. So, these things do need to be done at multiple places. And I think the surprise will be that this won't be that expensive to do. What I figured out is that you can do this with the kind of research funding that neuroscience labs are currently able to do. What are we doing? We divide the brain up. So what's the technique? You inject a tracer, and there's a variety of tracers. Each of these tracers can transport retrogradely or anterogradely, which means it can be taken up by a synapse, climb up along the axon and go back to the cell body, or be taken up by the cell body and then transport back along the axon out to the synapses. Now that's a caricature because complex mixtures of these things happen with any real tracer. But what we are doing is we are saying that we can do a fixed number of injections in the brain. I'm going to show you what those results look like. And again, we have brought slides, so you can take a look afterwards. So let's say we have an injection budget, 250 injections. With each one of them will be done with 4 different tracers. So we now have to place those injection targets in the brain. So we grid the brain up. And this is Pascal, who's sitting here somewhere, basically did a sphere packing problem to divide the brain up into a little grid. We tried to kind of respect neuroanatomical boundaries. This is a hard task because mice brains actually vary individually. You can't really do MRI-guided stereotactic neurosurgery as you would do for humans. So, the injection placement won't be perfect. But we will do lots of injections. So even if the injection placement isn't perfect, we get a bit of a random grid in the brain. We will still have good coverage throughout the brain.
So why 215? This is what you could do? Or
why 250? I just picked a number out of my hat. Yeah, it is resource constrained, and it will increase over time. But also remember that there'll be some amount of correlation length. So we don't need to go with an infinite number. That's kind of the idea.
But we don't know what's the right number.
We don't know. Okay, here's a little pipeline video. I don't know how much of this I want to show you, but maybe quick. Okay, so sectioning. This little floor plan of the lab. That's a critical step. So tape transfer assisted cryosectioning. Yeah, sorry.
I don't understand. The 215 question. Are you saying that you're dividing the mouse brain into 215 regions, and then looking to see which region communicates with which region?
What I'm doing is I'm taking n, which could be 250.
Yeah.
I'm placing n points in the brain, in a three-dimensional model of the brain, so that they are roughly equidistant from each other. For each point, I'm going to do one injection in one mouse.
Yeah.
Okay. Once I inject, that there'll be a bolus of tracer that will be taken up by neurons or synaptic processes there. It will be transported then. So I will show you what this looks like. It will be transported to other parts of the brain. It will not transport to every part of the brain. This projection pattern will be patchy. So from a given brain, we will learn how one region projects to all other regions or gets inputs from all other regions. So that's what's being done.
Resolution
But
targets.
this is like a very, very coarse level. So you're going to end up with a 215 by 215 matrix?
No, we are going to end up with a three-dimensional data set for each injection in which individual neurons can sometimes be seen. So it's a way of labeling the neurons and then observing the neuronal morphology to the extent that there are spatial correlations. I don't necessarily have to label every neuron individually. I should be able to label a statistically representative neuron in a region. So that's sort of the idea.
So it's 215 sources, but that may be more refined.
Well, the individual images have high resolution. They are micron level resolution. So you can, even though my injection is large, you can see lots of neurons in that injection region. You can potentially, you can't trace individual fibers, but you can trace whether this group of neurons was talking to another group of neurons. I will show you some figures.
So how does the number of injection points relate to the 468 regions?
Well, it's chosen to be roughly commensurate, right? So
But you hope that they're randomly spread among those 468, or are you aiming for one region?
Can't aim for, so some of those regions are really tiny, and they will have to be targeted later using genetic targeting. Some of the regions, one I will show you, is nice. And let me show you an example of how we can draw conclusions. Yeah.
How do you put together the data from different slices?
The stacks have to be. Let me come to this, because I will talk about it. So this is, by the way, I don't know how to pause this. Otherwise, I would pause it for you. That's actually a nice slide showing. Okay, just to give you a sense of what it looks like inside the lab. So those are multiple sections per slide. They get stained, so they are robotic devices essentially. So this is all done by hand in a typical neuroscience setting. But if you go to a hospital, of course, you'll find these machines to do histology. So you're basically taking histology machines and using them. We are following the Allen Institute here. You can see a brain laid out. That's a stack of slides. Each one of those boxes contain 2 brains if you want. These things get scanned. That's a deck of slides. There are these things which are very expensive instruments to scan a deck of slides, and then you have to kind of do some. That's Alex who's been helping out. We have to do data management and so on and so forth. Blah blah. We will eventually release this data on our website, but I don't want to talk about the limbs too much. We have to manage all this information, so we have a laboratory information management system. That's a screenshot of it. So we end up with a bunch of brains. What does the data look like? So let me show you what the data looks like a little bit so you can get an idea. So this is my cue to go to browser, which I have carefully opened and I hope will show. Now, remember, these are very multi-resolution images. It's very difficult to get a sense of it by looking at a small window. You can look at only one resolution. That's why I brought this large printout. You can start getting a sense. And what I've just told you is you need a 3-story big printout to really, although you will lose you lose light microscopy. We need at least a 10 times bigger printout to saturate the resolution we have in our lab. So you'll get only a crude idea by looking at what I'm showing you. The images themselves have a lot more resolution. So let's stare at this particular image for a little bit. This is a mouse that was injected with a fairly large bolus of cholera toxin tracer, which is roughly a retrograde tracer. It was injected in the caudate. So the injection, this is a very large injection actually. It's placed in the caudate nucleus. This section is close to the caudate nucleus. Not all of this region has been injected. The tracer has transported after being taken up by the processes. But what I want you to look at are these little bands here, which we can zoom into. Those are so-called layer 5 pyramidal neurons, which are projecting to caudate. Okay, so these neurons are sending processes here. The tracer was injected here, got retrogradely transported, and went here. This is a laminar path. Sorry.
So it's key to say where the tracer is taken up and the terminals.
In this case, ideally, these things have not been sufficiently empirically well studied. I can't, guarantee that, but that's the idea.
This passage along the axon is the only mechanism by which these tracers can be transported around the brain?
Correct. For long-range transport, correct. Active transport along the axon, which is why you can't do it in the dead brain. Diffusion is very slow. Now, something very interesting is going on. So we have injected tracer here, but there's label showing up here, there's label showing up here. What's going on is that the tracer is being transported back to these neurons, but these neurons send collateral branches over here. So actually what you're seeing here is not fibers crossing over like this, but in fact collaterals coming down from these guys down here. And these neurons which are labeled, in fact, send collaterals here, and they are picking it back up and going there. I think I have a slightly better zoomed in version. Okay, so you can see actually individual cell bodies labeled. So that doesn't look too good on the screen. I do have a different tracer injected, which maybe, so in this case, a rabies virus was injected also in the caudate. This is a retrograde rabies virus from Ed Calloway and Ian Wickersham. And you can't see it, unfortunately. It was injected here, it got transported up. These are the neurons which send projections here, And also these neurons send some projections here. Let me see if I can zoom in for you. Yeah, these are the neurons on the other side, right, that are sending fibers down to the caudate where we injected, and these neurons are sending projections directly.
So how do you know which ones are directly and which ones are indirectly?
No. They're all sending projections directly. The one is collateral, and the other, they're both being done directly. I don't want to belabor this, but you can sometimes observe individual neurons and draw conclusions about that. I don't have much time. I did want to talk about something else. So we have to do registration. Let me talk about that separately. I wanted to quickly touch upon, here's a mouse from Josh Huang with all the neurons with this GAD2 enzyme labeled in their nucleus. And this enables something nice. So Swagatam, who is sitting here also somewhere in the audience, developed a little cell counting algorithm where you can basically go in and pick out individual cells. I've shown you a little bit of a tricky portion where the cells are coming close together. But simple things like how many cells are there in the brain of a given type, we don't know. So that kind of thing, I think we'll be able to do. Can I take 5 more minutes to talk a little bit about? Okay, I wanted to very quickly touch upon what you could do when you have data sets like this. And I don't have my data set yet. I'm showing you the innards of the project as it is going on. I'm sorry, but we do have a data set from the Allen Institute, which is a gene expression atlas. So what they did is for about 20,000 protein coding RNAs, they did in situ hybridizations in these 56-day-old C57 black mice, and then quantified where the genes were being expressed. They then compiled that, they registered those brain sections together. We have to do the same sort of thing into one 3-dimensional data cube where each voxel now has a vector of gene expression, right? That comes by registering together many different brains together. Each one brain has only one gene being studied, actually 5 genes in different series. And this is the maximum intensity projection for one gene. So this is a particular gene called PROX1, and you can see that it is highly expressed in the hippocampal formation. So we took this matrix, which is voxel by gene, and did a singular value decomposition on it. And we did that to discover whether it was of low rank, whether it had structure in it. And not surprisingly, it had structure. Those of you who are familiar with singular value decompositions or principal component analysis will recognize these plots. What was remarkable about this data set is that we needed about 271 modes in order to capture 90% of the variance. So this is a microarray data set, but this is very different from the kind of microarray data sets people typically get by manipulating conditions or by getting cell types, because when you do principal component analysis on those data sets, you get a few modes, order 10, that capture 90% of the variance. So this is a really high dimensional data set. Now, if you look at the projections onto principal component space, you find out that different brain regions separate out. So that sort of striate, this little part here is cerebellum. If you want, the gene expression vectors are characteristic of brain region. After having thought about this for a while and done some analysis, we have come up with a little model of what is generating these correlations. And this is a very simple-minded model, but I think is essentially, it looks consistent with all our observations. And it is a model in terms of underlying cell types, which are hidden from us, which we don't know. Each cell type, so the brain has thousands of cell types probably. Each cell type has its own gene expression vector. When you look at a voxel in the Allen Atlas, what you're getting is a linear superposition of all the cell types. Because it is being studied one gene at a time, the cell types have not been specified. The matrix, the expression energy at a given voxel and for a given gene G is really a sum over the expression profiles of all the cell types, T, multiplied by how much of that cell type you have in that given voxel, right? So this is kind of a hidden variable model. It's a linear model, but I think is essentially accurate. And then there are residuals. And you can in fact fit this model because thanks to Sasha, Nelson, and others, we have the gene expression vectors of certain cell types. So Pascal went ahead and did that. And here's the cell type, which is the medium spiny neuron, which lives in the striatum. First of all, we do a projection, which is basically take the cosine of the angle between the cell type vector and the gene expression vector in a voxel in the Allen atlas, and we look at that cosine. So those vectors live in 3,000 dimensional space. So by chance, their cosine would be 1 over square root of 3,000, which is small. But the striatum, for example, shows large projection. It's not a perfect correlation. It's not a 90% correlation. It's only a 30% correlation. I've read some stories on Wikipedia that the striatum has 95% of its neurons are medium spiny neurons. If that were true, I would expect this correlation to be much higher. So that story may or may not be true. I don't know. But we also fitted the model that I showed you, this linear model. And since there's a positivity constraint, some of the coefficients become 0. So in fact, you can see that according to the model, the medium spiny neurons should live in the striatum, which is very happy. And what you can do is you can also estimate dimensions. So if you go from voxel to voxel, your vector changes a little bit. So if you take all of caudate, you can ask in what dimensional space does caudate live in terms of these gene expressions. And that gives you sort of a lower bound on the number of cell types that are living in caudate. Why does it do that? Because if there was only one cell type, or if there were 10 cell types, but they all had the same spatial profile, then they would actually show the same gene expression vector. But they don't. These gene expression vectors change. So to the extent that gene expression defines cell type, which it probably doesn't, but to the extent that it does, calculating the dimensionality, estimating the dimensionality of these voxels gives us a lower bound of the number of cell types that live in caudate. So here's something that I've inferred from the Allen data set that, I don't know anything about cell types. I'm making inferences. So I won't bore you with the details. The blue curve is sort of, this is a histogram. There's a statistical procedure. The peak is sort of our best estimate of the dimension of the vectors in caudate. It's a small number of dimensions. The good thing is when we remove the medium spiny neurons, the dimension estimate falls by 1, which is what you would expect, but it doesn't go to 0 actually. Same thing for cerebellar granule cells. We did try to match up with the reactome data set. That didn't work very well. We can cluster the gene expression vectors and recover classical neuroanatomy. I want to thank the NIH and NIMH for supporting this project and Keck Foundation for initial support my professorship. Cold Spring Harbor Lab, I do get funding from the Simons Foundation for some of my work. I want to thank, there's a lot of people involved, people working very hard in the lab. I don't want to go through everybody in detail. Vadim Pinsky has played a very important role. He's a bioengineering graduate student, but system engineering means getting these pieces and hooking them together. This is not consumer electronics, so they don't always play nice. So you may have to rejigger the pieces to put them into a pipeline. So Vadim has been very helpful with that. And a lot of collaborators. And in fact, now that we have set this pipeline up, people like Bruce, Jin-Young Lee, others are approaching us and we are starting collaborations. I want to end my talk. I will take a liberty here. Many of you have seen this book. Please raise your hand if you know where that phrase is taken from. What mad pursuit? Thou still unravished bride of quietness, the foster child of silence and slow time, sylvan historian who canst thus express a flowery tale more sweetly than our rhyme. What leaf-fringed legend haunts about thy shape, of deities or mortals or of both? In Tempe are the dales of Arcady. What men or gods are these? What maidens loth? What mad pursuit? What struggle to escape? What pipes and timbrels? What wild ecstasy? Now, I wasn't just impressing you with my memorization skills. I put it up for a reason. Okay? So that's on a Grecian urn from John Keats. That's the first stanza. I memorized the poem, the first stanza I memorized while on a flight took me about 15 minutes or so. Now, 400 characters in this first verse, that's about 2,000 bits. Try memorizing 2,000 bits in 15 minutes. It can actually be done. There are certain people who there are these things called memory Olympics. There are 2,000 characters in the poem, which I also know by heart. That's 10,000 bits. Try memorizing 10,000 bits just as a bit string, right? Now, the world memorization record for bit strings is 4,000 bits in 30 minutes. How is it done? It's actually done by using space and journeys in space. So there are these concepts of memory palaces, which was known to the Greeks, and people have developed it. So memory doesn't work by just taking bits and putting them down in individual synapses. It's highly structured. If you want to memorize a long string of bits, you have to take advantage of the structure that's present in your brain, evolved through time, that helps you navigate space. That's you memorize bit strings. And the reason I put that up there is to show you that there is indeed structure in the brain to be understood. It's not just about understanding synaptic weights. Thank you.
Okay, why don't you take a few questions here, then we can take more outside. Or we can...
You mentioned convergent evolution. I wonder if you could explain what that is.
Okay, so I'll explain with an example. So the one of the best examples is the cameral eye in the octopus and in the human. So a cameral eye is something that's like a camera. It's a little lens. It's got a little photographic film at the back. The octopus has it, we have it, but our common ancestor doesn't have it. They have a common ancestor, the Ur-Bilaterian, whoever that was, a little worm, creepy crawly, didn't have a cameral eye. Did probably have the genetic, the molecules to make opsins and so on and so forth, pigments, but did not have the cameral eye. So it was evolved separately in two lineages, same design. And the idea is that if you see the same structure evolve independently, then it is sort of function constraining form. And what I would posit is biology has a lot of information, a lot of form. How do you filter it? Well, convergent evolution is a filter. It is a function filter.
So something like myelin is not found in C. elegans or Drosophila. So you would consider that not convergent.
Could you repeat the question?
Question is, myelin is not found in Drosophila and in C. elegans. So how does this fit into convergent evolution?
No, but there is a form of myelin that's found in invertebrates. Crustaceans have a form of myelin, and it's unrelated.
I do not.
It's not in the fly or the worm.
Wait, to have convergent evolution, you have to have it. You have the trait in at least.
But my example is an example of convergent evolution because crustaceans have a form of nerve that has something homologous to myelin. And so it may not be a model organism, but it is an example of convergent evolution.
Yeah. I mean, I don't know enough. I'm not an expert in this particular example, so I won't comment on it. But my point is that among the infinite variety of fact in biology, if we pick those which replicate themselves across the taxonomic tree, not being present in the common ancestor, we have discovered engineering principle, some design principle or invariance. And I'm positing that, for brain circuits we can do that. There are examples for microcircuits probably, but it would be great if we can do it for the sort of this meso-level circuitry, really help us understand, I think, how the brain works. How is the octopus brain convergent with our brain? That'd be the great one to study, because the octopus has fairly developed cognitive skills.
Can you say, perhaps again, but I didn't get it, why 1 micron resolution is the right?
I did not say it is the right one. It is the optical.
Why you chose it?
Optical microscopy. So
it's the best you can do, or it's the and why is it good enough?
It is good enough for certain purposes. I think that it uncovers neuronal morphology at the level of long-range projections, which is what we are interested in. So it does uncover that. It is adequate for that. It does not tell you which neuron is connected to which neuron. You have to do both electron microscopy and electrophysiological and other more direct methods. Even with electron microscopy, it's hard.
Single neurons?
Um,
I mean, wouldn't that
you
be-
can look at single neurons using optical microscopy. There's no problem with that. You can look at the morphology. Remember this remarkable morphological feature of neurons. Very, very long processes. You can see that under an optical microscope. So that's what we are kind of focusing on here. So it's one of the levels at which the system has a self-consistent structure.
So, sort of to understand better how far you've gotten, you've done some number of brains so
Yes.
far, and Alan has one. Just say a little bit more about what's
Allen Institute is also doing what they call a genetically defined connectional atlas. I don't know the details of their project. The gene expression atlas that they generated, that's thousands of mice. In our project also, over time, there'll be thousands of mice. So on scale terms, we'll be similar. Where are we now? I have to disclose trade secrets. We have hundreds of brains scanned, but for the first project here, we want to order 1,000, but that now I have to kill you.
understand that for each, it takes one mouse to...
One injection per mouse. We are doing one injection with one tracer per mouse. So for any injection site, we are doing 4 tracers. That's 4 different mice. And then, as you can see, even one injection produces a rich projection pattern. I did bring some of our project slides, so any of you who have expertise and want to look at how good our material looks, we are open house.
But so that's 1,000 mice. So with 1,000, you won't start to be able to even ask about sort of the variance from one mouse to another.
In the project over over a multi-year period, we do plan to do n equal to 6. So I did very much recognize. So first of all, with multiple tracers, you already get multiple n. But one has to study the variance. I completely agree. It will just take a little time. We are completely limited by our resources. We can, and I don't want to get into political trouble here, but the sort of amount of knowledge gathered per dollar will be more efficient than certain brain imaging techniques by far.
Partha, your traces are taken up indiscriminately. Are you doing any cell-specific?
We are going to move there, right? So this collaboration with Josh that's starting with... So the way I see that is there has been this sort of, a bit of like a cell-type Kool-Aid in the sense that...
Probably should repeat the question.
Okay. The question is, the tracers in our project are taken up indiscriminately. Do we have any plans to specialize them to individual cell types? The answer is yes. But I would like to structure that by not just going for cell type per se, but thinking about what we know about the architecture. We know that there are these neurotransmitter families, these receptors, and we want to make some logical choices. So one of the things that we are going to do next, hopefully, is collaboration with Josh Wang. He's labeling interneurons. So we are going to try to specialize to classes of interneurons. So that's giving you the negative.
What's an interneuron?
I should not have said interneurons. I said the wrong thing. I'm sorry. My 2 hours of sleep are catching up with me. He's labeling GABAergic neurons. So Camillo Golgi in his paper pointed out there are 2 classes of neurons, interneurons and projection neurons. Interneurons stay local, projection neurons go far. Many interneurons are also inhibitory neurons, but not all inhibitory neurons are interneurons. Josh has got these set of mouse lines which label specific GABAergic cell types. So that so we will refine over time. Yes.
I'm going to suggest we continue up here or out by the line. Thanks very much.